Welcome back. Welcome back. This is the review video for control of microorganisms, which is not such a big topic in this class. I mostly want you to understand some concepts behind it that you can apply to any control method. So not so much teaching you a bunch of random things as teaching you the concepts you use to analyze a bunch of random things. So here's where we start, and this is a topic we hit a little harder in the next one, infective dose. This is a minimum, just the smallest number of microorganisms of any particular type that can give you a disease. So the smallest number of virions that can give you COVID, or the smallest number of virions that can give you norovirus. It's not usually one, because one virion is just likely to get stuck in some mucus and not get to a cell. So there have to be some probabilities at work here. And infective dose ranges from around 10 at the smallest estimate for something like norovirus or certain E. coli species that do a lot of damage to people, all the way up to 10 to the 11th power, which is 100 billion cells for weak strains of Vibrio cholerae that causes cholera. So that's a huge difference. If you're looking at a huge infective dose like that, it means you can drink a huge number of those cells and not get cholera. But most infective doses are likely to be a lot smaller. And again, we'll look at this in more detail in Lecture 10, but the point is that this is the goal of disinfection and control processes. We're always trying to get below some infective dose. We're just trying to make things safe by killing microbes. That's all. So just make sure you know what infective dose means. There are big words. We go for bactericidal. So what does cidal mean versus bacteriostatic? What does sterilization mean instead of disinfection? And both of these words are hugely important. Sterilization and sterile refer to killing every single thing, making every virion inactive, making every cell dead, making every endospore inactive. Zero, absolutely zero of anything left. That's sterile. If there's a one of something left that's still alive or infective, it's not sterile. So disinfection usually involves like reducing the numbers a lot, making them safe, but not making something sterile. And we use disinfection informally to mean like trying to kill stuff, but sterilization, that is serious, and that only means the one thing. We looked at modes of action, and those, again, look at what they are, and remember to compare them to the modes of action of antibiotics, because modes of action of disinfection technologies and sterilization technologies and all that, those are really, really broad and really, really, broad acting. Like heat damages every protein in a cell versus an antibiotic that interferes with one enzyme in a cell. Let alone all the structural proteins or all the signaling proteins that get ignored by an antibiotic. Heat damages all of them. So it's very different. Kinetics and decimal reduction time. That's another thing we looked at. Make sure you can calculate a decimal reduction time. And again, decimal reduction time equals the total time divided by the number of decimal reductions. That's all it is. You might see a definition involving logarithms, and if you boil that down, this is what it means in English. So be able to calculate that. part of why we do this is to help us remember the 3 C's of disinfection. Remember what they are, and absolutely remember what they are, and absolutely know how to apply them. And definitely one of them doesn't really start with C, and that is the most important one for you to remember, because that's the one most people forget. Contact time is not something people necessarily think about, but everyone should. If you put even bleach, which is my favorite thing, if you put bleach on a surface to kill endospores, it doesn't work instantly. It takes time to get into them, and each one has a certain probability of dying in a certain amount of time. And so it takes a certain amount of time to kill them all. And it may be short, like a minute or 5 minutes, but it's not instantaneous, and you can't pretend it is. So if you see someone spray a disinfectant and then immediately wipe it away, they're not doing it right. And if you see them wipe a surface with Cava 1 or Cava Wipes 1 or Santa Cloth, even the maroon kind, the plum kind, and then immediately dry it off, they're doing it wrong. Make sure you follow the instructions or it won't work. Okay, and then methods. Methods we talked about involved moist heat, which is useful because it carries a lot more heat than dry heat does. And so that involves boiling water, which is a very common disinfection method, and steam, which is very common in professional settings, where you can control the temperature and pressure of steam in an autoclave. So make sure you know what both of those are. Incineration is dry heat, and it's at a high enough temperature that it works, and it just sets everything on fire, and nothing can survive being on fire. Filtration is a way you can actually sterilize stuff routinely in a laboratory. You can make filters with small enough little holes that they block endospores or even virions from getting through. Cold, what does that do? Think about the effect cold has on a cell and why cold is able to protect food from having stuff grow in it. We also looked at radiation, ionizing, and UV. And what else did we look at? Bleach, alcohols, like for hand sanitizer, it's good to know, and surfactants. So regarding these methods, make sure you know their modes of action, what they are, like know that steam is referring to an autoclave, know what an autoclave is, and know whether they are capable of routinely achieving sterilization or not. Like, would somebody use them primarily to sterilize something? Or would someone be able to use them to sterilize something in an easy amount of time with, an easy situation? So for example, boiling water, no. You'd have to boil water for several hours to be confident you'd sterilize it. No one's going to do that. Whereas incineration is always going to sterilize everything, because even prions are going to become carbon dioxide. You can sterilize things using radiation and bleach, but not alcohols. So, just look those things up in the slides, and if they're there, learn them. And that is it for control of microorganisms. I'll see you at the next video.